In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.